Just like any skeleton that is present in the living organisms, cell also has a skeleton. Therefore, we refer it as cytoskeleton. Normally, जो सेल का थ्री डायमेंशनल स्ट्रक्चर है उसको जो है इंटरनल सपोर्ट जो दी जाती है दैट इज फ्रॉम साइटोस्केलेटल एलिमेंट्स. इस साइटोस्केलेटन के कंपोनेंट्स के हवाले से अगर हम बात करें तो तीन मेजर कैटेगरीज हैं. द फर्स्ट वन आउट ऑफ दोज इज माइक्रोट्यूब्यूल्स, and we will be talking about microtubules, their function in detail, and then we will talk about the other elements like intermediate filaments and then uh, microfilaments. If we look at the major functions of cytoskeleton before we proceed further for detailed study, then we can broadly classify that five functions are mainly performed by the cytoskeletal elements. If you look at this shape of the cell, that is a filamentous cell, और अगर आप जो है वो circular cell को देखें, जो cellular shapes हैं, they are maintained by special arrangement of these cytoskeletal elements. How they are arranged, then that is going to determine the shape of the सेल. अगर हम सर्कुलर सेल की बात करें तो सर्कुलर सेल में जो है साइटोस्केलेटल एलिमेंट्स और मेनली माइक्रोट्यूब्यूल दे रेडिएट आउट फ्रॉम द सेंटर. अदरवाइज अगर आप एपिथेलियल जो कलमनर सेल की बात करें देन दीज माइक्रोट्यूब्यूल्स आर रनिंग पैरेलल टू द लॉन्गिट्यूडिनल एक्सिस of the cell. Then these structural elements are responsible for positioning of the cellular organelles. अगर आपको याद है हमने बात की थी that nuclear envelope is in continuation with the rough endoplasmic reticulum, then smooth endoplasmic reticulum, then Golgi complex. इसके अलावा हमने बात की that mitochondria are specifically arranged in those parts of the cell where energy is required more. Similarly, we can simply say that all the cellular organelles, they are present and they are arranged at a specific position within the cell along with the cytoskeletal elements. आप इसको इस तरह से समझ सकते हैं जैसे बुक शेल्फ्स में बुक्स को अरेंज करने के लिए स्पेसिफिक शेल्फिंग हुई होती है, उसी तरह से विद इन द थ्री डायमेंशनल सेलुलर शेप सेलुलर स्ट्रक्चर में अंदर वाली साइड पे साइटोप्लाज्मिक वाली साइड के ऊपर जितने भी अरेंजमेंट्स हैं इन सेलुलर ऑर्गेनेलिस की दे आर बिकॉज ऑफ द साइटोस्केलेटल एलिमेंट्स. Then we spoke about the intracellular transportation of, like, for example, secretory vacuoles की बात की थी that they move up to the plasma membrane, और endocytosis के result में endosomes की बात की थी, they move towards the center of the cell. This movement is involved the utilization of these cytoskeletal elements like microtubules and some other cytoskeletal elements as well. Just like we use roads for communication. So cell के अंदर जितनी भी transportation होती है, movement होती है materials की, that needs the cytoskeletal elements. Then for the cellular movements, like for example cilia and flagella, locomotory organ के तौर पे ये जो है elements function करते हैं. And if we look at the cell division or cell division में भी in particular अगर हम देखें माइटोसिस के डिफरेंट फेजेस में जो स्पिंडल फाइबर्स हैं दे आर साइटोस्केलेटल एलिमेंट्स. उसके बाद अगर साइटोकाइनेसिस देखें, साइटोकाइनेसिस इज बिकॉज ऑफ these cytoskeletal elements. So in other words, we can say that cytoskeleton is not only providing the support function, it has to perform number of other functions within the cell. Starting with the microtubule, as we can see that the name is microtubule. These are tubular structure. If you look at this picture, these are tubular structure which are made up of by different filaments which are aligned side by side, and each filament has repeated units of alpha and beta subunits of the protein tubulin. अगर हम लेंथ के हवाले से बात करें, the length of these microtubules is variable depending upon the size of the cell. फॉर एग्जांपल एपिथेलियल सेल की बात करेंगे तो उसकी शेप जो है कॉलम की तरह की है. अगर अगर आप एक्सोनल सेल्स की बात करेंगे देन एक्सॉन सेल बॉडी से जो ओरिजिनेट होती है its length can be quite much long, starting from the backbone up to the tip of your finger. So you can just imagine how long can this axon be. So this length of microtubule is variable and depends changes from cell to cell. If we make a cross section of this microtubule, then this is what we can see under electron microscope, that these structural subunits they are arranged in a circular fashion around a central cavity, and 13 such subunits are visible. If we talk about the size of the microtubule, from end to end, the diameter is 25 nanometer. And if we look at the thickness of this wall, the thickness is 5 nanometer, leaving behind 20 nanometer size of the cavity which is present within this tubule. When we look at uh, these individual filaments, like if I draw a line here, this individual filament is referred as protofilament. Microtubule is made up of tubulin protein, is number one point. There are two types of tubulin protein, alpha and beta tubulin protein, which makes up the heterodimer. This is the second point. And then the third point is that these heterodimers, they polymerize in a lengthwise manner to make up a single filament, which is called as protofilament. 13 such protofilaments, they come side by side and they make up the microtubule, which is the which is the component used for multiple functions in the cell. Keeping in view the polarity of or keeping in view this structure where we have the first protein subunit of alpha in at one end of that this protofilament and beta on the other end of this protofilament. All these filaments, 13 protofilaments, they all have alpha subunit on one side and they have beta subunit on the other side of the filament. Because of this property, we can call that these protofilaments are polar because they have one pole which is having alpha subunit, the other pole is having beta subunit. Besides this, we call the beta subunit carrying end as positive end and alpha subunit carrying end as negative end. Because of these alpha and beta subunits on opposite ends, overall microtubule becomes a polar structure that its one end has all the beta subunits and is referred as positive end, and the other end has all the alpha subunits and is referred as negative end. एक और जो प्रॉपर्टी है पॉजिटिव एंड की, that polymerization will always occur on the positive or plus end of the microtubule. So the growing end is going to be the plus end of the microtubule. यहाँ तक बात क्लियर है जी?
Yes,
Then
sir.
Yes.
we
Yes, sir.
then we come up. Okay. All right. Then we come up with to the different microtubules which are present in the cell. Like if I look at this picture, you can see this blue colored rod shape structure. This is one microtubule, and the in parallel there is second microtubule over here. There are some specific proteins which are present in between different cytoskeletal elements. If this is one microtubule, one protein is going to be attached with this microtubule and its tail is protruding out. The function of this tail is to interact with the second microtubule so that the two microtubules are held at a specific distance from each other, and at the same time the spatial organization of microtubular structures can be maintained. आप इसको जो है हॉरिजॉन्टल स्पेसर्स के तौर पे कंसिडर कर लीजिए। आप इसको जो है वो आप कंसिडर कर लें कि such proteins are responsible to keep the different microtubules at a specific position arranged within the cytoplasm. Such proteins, they are called as microtubule associated proteins or MAP proteins, which are of multiple types, but we will only take the example of certain classical maps. MAP2 is one of the example, which is responsible for acting as a cross bridge between microtubules and other cytoskeletal elements to maintain the parallel alignment. उसके बाद अगर हम इनका फर्दर रोल देखें मैप्स का, दे आर इन्वॉल्व इन स्टेबिलिटी of microtubules and they promote its assembly for the reason कि अगर आपने कभी एक्सपीरियंस किया हो कि कुछ ब्लॉक्स को स्टैक अप किया हो वन अबव द अदर, देयर कम्स अ टाइम जब स्टैक जो है स्टैक इतना टॉल इतनी हाइट तक चला जाता है that it becomes difficult for the stacks to remain stable, and it can go drop on one side or it can drop on the other side. So the same thing may happen with these protofilaments, that they can go up to a certain length, and if they are not supported from the lateral sides, there is possibility of collapse because of extra long microtubules can no more be stable without the support of these microtubule associated proteins. These microtubules, as I earlier mentioned, they are responsible for arrangement and structural support and act as organizers. If you look at this mitochondrion is basically held at this place because of this microtubule or other cytoskeletal elements. If you look at this rough endoplasmic reticulum, again it is associated with certain cytoskeletal elements. And if you use certain drugs that depolymerize the microtubules, then this would look like इस जो arrangement है in cytoplasmic organelles की, it will be lost, and it will be like due to gravity, these organelles can be dropped at a common point within the cell, and it looks like आपने किसी बैग में जो है जो शॉपिंग बैग में कुछ जो है वो वेट कैरी कर रहे हैं, and all the weight is towards the bottom of the shopping bag instead of having this three-dimensional arrangement of the organelles. So this is another function of microtubule, to provide structural support and act as an organizer for the cytoplasmic organelles. Regarding intracellular motility, as the best example for motility observation is the nerve cell because of its long axons. And you might have studied in physiology that certain vesicles, they are transported up till the end of these axons because there is a presynaptic membrane and postsynaptic membrane, and there the neurotransmitter has to be released. Just consider this as an example of that nerve, and this vacuole that has to go up to the end of this axon to release the neurotransmitter. How this vacuole will move? This vacuole will walk around the cytoskeletal element, that is microtubule. Now keep in mind that this vesicle is moving away from the cell body or away from the nucleus towards the plasma membrane, and this end of the microtubule is basically plus end, and the end which is towards the nucleus is the negative end. So when the vacuoles are moving from minus towards plus end, this kind of movement is called as anterograde movement. That means the walk of the molecule, or the walk of these vesicles is towards the plus end of the microtubule or is towards the plasma membrane. And if we talk about some opposite direction of the movement, that something entered into the cell and it is moving towards the main part of the cell, then this kind of movement is called as retrograde movement, because this movement is happening from positive towards minus end. This is one best enlarged picture or example in which the organelle is being transported along this microtubule with the help of such some motor proteins, and that movement is towards positive end from minus end. और ये जो black color की आपको lines visible हैं, these are basically the map proteins which are keeping these associated or these parallel microtubules at a specific distance. Besides which, there are certain other elements like intermediate filaments. उनके साथ भी जो है वो स्पेसिंग को मेंटेन करने के लिए माइक्रोट्यूब्यूल एसोसिएट फिलामेंट्स जो है वो अपना रोल प्ले करती हैं. देन कम्स अप, ओके, यहाँ तक कोई बात पूछना चाहते हैं तो पूछ लीजिए. ओके जी. Then we talk about certain motor proteins which are responsible for um, intracellular mobility or cellular movements. As I mentioned earlier that there is a minus end and plus end of microtubules. So some of the movement is from minus towards plus end, and sometime movement is from plus to minus end. Before going into detail of these motor proteins, I just want to recall your previous knowledge of actin and myosin proteins, and you might have studied myosin as a motor protein. Just keep in mind about the structure of the myosin, its head region, heavy chains, light chains, and we are basically going to talk about something similar with the myosin proteins. When we talk about cellular movement, the movements, intracellular movements, then the motor proteins can be divided into three superfamilies. One is kinesin, the second one is dynein, and the third one is myosins. Myosins जो हैं, they interact with the microfilaments like actin, whereas kinesin and dynein, they interact with the microtubules. And out of these kinesin and dynein, those motor proteins which are responsible for mobility towards positive end, they are called as kinesin, and those which are responsible for movement or transport towards negative end, they are referred as dynein proteins. And if we look at the structure of kinesin, this is one kinesin which is visible as a heterodimer having one head region, and this head region has a link with the stalk through a neck region. This is the neck region, and then there is a tail region with which the light chains are involved. So what we can figure out from this picture that kinesin has four polypeptides. Two are heavy chains, two are light chains. Heavy chains are making up a head region and a stalk region. Head regions are connected with the stalk region through neck region, and in the tail region we have the light chains which may show the variations. अगर हम kinesins के types देखें within any cell, there are almost 14 different types of kinesins which are present, and head region of these kinesin from one cell to the other cell, if you compare it, the sequence of the head region is more or less similar. On the other hand, when we look at tail region, then the sequence variations, the protein sequence variations are visible. उसकी वजह क्या है? That this head region has to walk along the microtubule. If you look at this blue and this red part, these are basically these head region molecules, and you can see this head region is simply walking along the microtubule. And this picture, if you look at the top, it shows the path of the movement. This is one step, and if you look the next step, it is going to be over here from here to here. So in this way, the walk around along the microtubule is achieved by the head region, and microtubule are made up of alpha beta tubulin in any type of cell. So this can be one of the evidence that why the head region from different cells has no changes, is conserved in amino acid sequence, because it is going to always interact with alpha or beta tubulin. On the other hand, the tail region is responsible to lift or to attach with the cargo. अब cargo की definition क्या है? Anything that has to be transported within the cell. It is a mitochondria, it is a vesicle, it is a secretory vacuole. So this has to be held by the tail region. So that can be the reason कि हमारे पास variety of such transport cargo is available. So we may have different types of sequences at tail region. And all this movement needs energy, and that energy is derived from ATP. This is just like the movement of myosin head when it is involved in muscle contraction. जी, any question?
सर कार्गो के बारे में एक दफा दोबारा बता दे।
जी, अभी इस पिक्चर में और क्लियर होगा, मैं अभी दोबारा से बता देता हूँ। ओके, कार्गो जो है, नाउ इफ यू लुक एट दिस पिक्चर, जिसको मैं एनसर्कल कर रहा हूँ, दिस इज अ वैक्यूल दैट इज बीइंग ट्रांसपोर्टेड विद इन द सेल. सो दिस इज अ कार्गो. यहाँ पे देखिए, दिस इज अ लाइसोसोम, दिस इज अ कार्गो, दिस इज अ वेसिकल, दिस इज अ कार्गो, दिस इज अ माइटोकॉन्ड्रिया विच इज बीइंग ट्रांसपोर्टेड, दिस इज अ cargo. Similarly, this is something VTC. I don't exactly remember what is this. अभी देख लेते हैं. This is transported towards the Golgi complex. Again, this is a cargo. So anything that has to be moved within the cell, that is called as cargo. आप जब अपने घर से मूव करते हैं, हॉस्टल आते हैं, तो आपके साथ जितना सामान आपके लगेज में होता है, that is your cargo, which you are transporting from your house to hostel or your hostel to your house. Okay, the kinesin is meant to move towards positive end or towards plasma membrane of the cell. Dynein, cytoplasmic dynein, has to perform opposite function. Cytoplasmic dynein again have the same regions, that it has a heavy chain, not one but two heavy chains. Then it has some intermediate and light chains which are connected with the head region through a stem, and there is a stalk which interacts with the microtubule on the this end. Unlike to the kinesin, जहाँ पे हमने बात की 14 different types की kinesin मौजूद हैं, there are only two types of dynein which are present within the cell, and out of these, only one type is performing most of the transport function. And out of this structure, the large globular head interacts with the microtubule, whereas the intermediate and light chain or tail region is going to interact with the cargo. So there is a possibility that within one cargo you may see dynein protein or you may see the kinesin protein, depending upon the direction of transportation. If transportation is towards minus end, then dynein is interacting with the microtubule. If transportation is towards the plus end, then you can see that kinesin, they are interacting with the microtubule. And by this way, these kinesin and dynein, as acting as a motor protein at the expense of energy which is derived from ATP, they are responsible to walk along the microtubules so that the transportation can be performed. Okay. Any question about material transportation? Okay. Then there comes a question that within the cell, how the polymerization of tubulin starts. The polymerization of tubulin starts because of certain specific centers. अब ऐसे नहीं है कि जो ट्यूबुलिन है at its own polymerization start कर देगी और its own जो है वो depolymerization start कर देगी at any point, any place in the cell. It is not like that. Within the cell, there are some points which are called as microtubule organizing centers or MTOCs. अगर मैं एग्जांपल्स की साथ बात करूं तो आपने यूकेरियोट्स में जो है सेंट्रियोल्स या सेंट्रोसोम्स का पढ़ा हुआ है। अगर फ्लैजेला या के हवाले से बात करूं, then you might have studied this क्लेमाइडोमोनास। और फ्लैजिलेट किसी और की एग्जांपल लें तो गालिबन यूग्रीना इज वन ऑफ द एग्जांपल। And every flagellum, it originates from a basal body or basal point. So these are two examples of microtubule organizing centers, which are the points from where the microtubules will originate. How it happens, if you look at this picture, you have this is the centrosome, and this centrosome is moving towards one pole. It has a pair of centrioles, and this pair of centriole, if you look at here, this pair of centriole is at right angle to one another, and these centrioles are surrounded by a cloudy mass which is called as pericentriolar material. This pericentriolar material is nothing different. These are the tubular molecules which are going to be used for polymerization so that the microtubule can be generated or can be elongated. Start of microtubule and elongation, these are two-step process. जो पहला process का step है that is called as nucleation. Nucleation में very first जो heterodimer है microtubules का alpha beta tubulin का, that comes and gets itself associated with the M tops. This is called as nucleation. Just like nucleus, न्यूक्लियस एक सेंट्रल पार्ट है सेल का, या किसी भी जगह पे अगर आप एटम में बात करें तो न्यूक्लियस की बात करते हैं, दैट इज सेंटर ऑफ द एटम। सिमिलरली यहाँ से पॉलीमराइजेशन ऑफ माइक्रोट्यूब्यूल स्टार्ट होनी है, दैट इज कॉल्ड एज the process is called as nucleation. Soon the nucleation has occurred, then polymerization starts and elongation of microtubule keeps on going. And this process keeps on going at unlimited time or as per cell's requirement. So there comes a time when we see that on one side of the microtubule polymerization is happening, and on other side depolymerization also starts. So this is possible, and that process when the depolymerization is happening at low rate and polymerization is happening at a higher rate, the net change will be extension in the microtubule filament's length. और अगर ये rate of polymerization or depolymerization equate हो गया है, then this process is called as treadmilling, and there is no net change in the length of the microtubules. माइक्रोट्यूब्यूल न्यूक्लिएशन के लिए जो बात हम कर रहे थे, न्यूक्लिएशन में जो फर्स्ट सर्कल ऑफ फर्स्ट पॉलीमराइजेशन ऑफ अल्फा बीटा सब यूनिट occurs, that occurs at us an other type of tubulin protein which is called as gamma tubulin. Gamma tubulin is responsible for nucleation of alpha and beta of nucleation of microtubules and attachment of alpha and beta tubulin proteins. So soon you get this alpha tubulin at the mTOX, then polymerization can start. Experimental studies have shown कि अगर हम इन gamma tubulin को antibody के साथ block कर दें, then there will be no point where the nucleation can occur. Then we will see that this microtubule growth is halted, and there is no extension, no growth of microtubule visible in the cell. Experimentally or within the cell, we can do some physiological changes which can introduce the assembly of microtubules or disassembly of microtubules. Like for example, the favorable circumstances or condition is 37 degrees centigrade temperature for assembly of microtubules, low calcium ion concentration, and presence of magnesium ion concentration. These are the favorable conditions for assembly of microtubules. uh, for the extension or for cellular purpose. And if we reverse the conditions, we decrease the temperature, increase the calcium level, or we use certain drugs which are referred as microtubule disrupting drugs, for example, colchicine, colcimide, nocodazole, they inhibit the polymerization of microtubule, and ultimately there is no polymerization towards plus end. इसके अलावा अगर आप कुछ ड्रग्स हैं लाइक विनब्लास्टीन विनक्रिस्टीन अगर आप इनको यूज करेंगे दे विल aggregate the tubular subunit, and these tubular subunits will no more be available to remain in the protofilaments, so they will go towards the depolymerization. And the last taxol is responsible for stability of microtubules. So both ends are met. There are certain drugs which introduce the depolymerization. There are certain drugs which inhibit the polymerization, and there are certain drugs which introduce the stability to the microtubules. So this is all about microtubules. If you have any question, you may please ask.